Okay, taking a little bit of a break from programming, I'm going to read over 4 quick web pages. This first web page is WebMD. Right here at the top, you can see the address if you want to look it up for yourself. And it's called Picture of Blood Human Anatomy by Matthew Hoffman, MD. So here you got a picture of a blood vessel and a lot of things that are inside the blood. Over here to the left, you see plasma. Down here, you see neutrophil. Up at the top middle, eosinophil, basophil, platelet, macrophage, lymphocyte, and then you've got the red blood cells here. Blood is constantly circulating fluid, providing the body with nutrition, oxygen, and waste removal. Blood is mostly liquid, with numerous cells and proteins suspended in it, making blood thicker than pure water. The average person has about 5 liters or more than a gallon of blood. A liquid called plasma makes up about half of the content of blood. Plasma contains proteins that help blood to clot, transport substances through the blood, and perform other functions. Blood plasma also contains glucose and other dissolved nutrients. About half of blood volume is composed of blood cells. Red blood cells, which carry oxygen to the tissues, white blood cells, which fight infections, platelets, smaller cells that help blood to clot. Blood is conducted through blood vessels, arteries, and veins. Blood is prevented from clotting in the blood vessels by their smoothness and the finely tuned balance of clotting factors. Blood conditions: hemorrhage or bleeding. Blood leaking out of blood vessels may be obvious, as from a wound penetrating the skin. Internal bleeding, such as into the intestines or after a car accident, may not be immediately apparent. Hematoma, a collection of blood inside the body tissues. Internal bleeding often causes a hematoma. Leukemia, a blood form of cancer in which white blood cells multiply abnormally and circulate through the blood. The excessive large numbers of white cells deposit in the body's tissues, causing damage. Multiple myeloma, a form of blood cancer of plasma cells similar to leukemia. Anemia, kidney failure, and high blood calcium levels are common in multiple myeloma. Lymphoma, a form of blood cancer in which white blood cells multiply abnormally inside lymph nodes and other tissues. The enlarging tissues and disruption of blood's functions can eventually cause organ failure. Anemia, an abnormally low number of red blood cells in the blood. Fatigue and breathlessness can result, although anemia often causes no noticeable symptoms. Hemolytic anemia, anemia caused by rapid bursting of large numbers of red blood cells called hemolysis. An immune system malfunction is one cause. Hemochromatosis, a disorder causing excessive levels of iron in the blood. The iron deposits in the liver, pancreas, and other organs, causing liver problems and diabetes. Sickle cell disease, a genetic condition in which red blood cells periodically lose their proper shape, appearing like sickles rather than discs. The deformed blood cells deposit in tissues, causing pain and organ damage. Bacteremia, bacterial infection of the blood. Blood infections are serious and often require hospitalization and continuous antibiotic infusion into the veins. Malaria. Infection of red blood cells by Plasmodium, a parasite transmitted by mosquitoes. Malaria causes episodic fever, chills, and potentially organ damage. Thrombocytopenia, abnormally low numbers of platelets in the blood. Severe thrombocytopenia may lead to bleeding. Leukopenia, abnormally low numbers of white blood cells in the blood. Leukopenia can result in difficulty fighting infections. Disseminated intravascular coagulation, or DIC, an uncontrolled process of simultaneous bleeding and clotting in very small blood vessels. DIC usually results from severe infections or cancer. Hemophilia, an inherited genetic deficiency of certain blood clotting proteins. Frequent or uncontrolled bleeding can result from hemophilia. Hypercoagulable state. Numerous conditions can result in the blood being prone to clotting. A heart attack, stroke, or blood clots in the legs or lungs can result. Polycythemia. Abnormally high numbers of red blood cells in the blood. Polycythemia can result from low blood oxygen levels or may occur as a cancer-like condition. Deep venous thrombosis, or DVT, a blood clot deep in the vein, in a deep vein, usually in the leg. DVTs are dangerous because they may become dislodged and travel to the lungs, causing a pulmonary embolism. Myocardial infarction, commonly called a heart attack. A myocardial infarction occurs when a sudden blood clot develops in one of the coronary arteries which supply blood to the heart. There's 2 more pages of this. Blood tests. Complete blood count, an analysis of the concentration of red blood cells, white blood cells, and platelets in the blood. Automated cell counters perform this test. Blood smear. It's not letting me highlight this. Blood smear. Drops of blood are smeared across a microscope slide to be examined by an expert in a lab. Leukemia, anemia, malaria, and numerous other blood conditions can be identified with a blood smear. Blood type. A test for the compatibility before receiving a blood transfusion. The major blood types are A, B, AB, and O and are determined by the protein markers or antigens present on the surface of red blood cells. Combs test. A blood test looking for antibodies that could bind to and destroy red blood cells. Pregnant women and people with anemia may undergo Combs testing. Blood culture. A blood test looking for infection present in the bloodstream. If bacteria or other organisms are present, they may multiply in the tested blood, allowing their identification. Mixing study. A blood test to identify the reason for blood being too thin or abnormally resistant to clotting. The patient's blood is mixed in a tube with normal blood, and the mixed blood's properties may provide a diagnosis. Bone marrow biopsy. A thick needle is inserted into a large bone, usually in the hip, and bone marrow is drawn out for tests. Bone marrow biopsy can identify blood conditions that simple blood tests cannot. And lastly, blood treatments. Chemotherapy. Medicines that kill cancer cells, leukemias, and lymphomas are usually treated with chemotherapy. Blood transfusion. A blood donor's red blood cells are separated from their plasma and packed into a small bag. Transfusing the concentrated red blood cells into a recipient replaces blood loss. Platelet transfusion. A blood donor's platelets are separated from the rest of the blood and concentrated into a plastic bag. Platelet transfusion is generally only performed when platelet counts fall to a very low level. Fresh frozen plasma. A blood donor's plasma, or liquid blood, is separated from the blood cells and frozen for storage. Plasma transfusion can improve blood clotting and prevent or stop bleeding that's due to clotting problems. Cryoprecipitate. Specific proteins are separated from blood and frozen into a small volume of liquid. Cryoprecipitate transfusion can replace specific blood clotting proteins when their levels are low, such as in people with hemophilia. Anticoagulation. Medicines to thin the blood and prevent clotting in people at high risk from blood clots. Heparin, enoxaparin, Lovenox, and warfarin, or Coumadin, are the medicines most often used. Antiplatelet drugs. Aspirin and clopidogrel or Plavix interfere with platelet function and help prevent blood clots, including those that cause heart attacks and strokes. Antibiotics. Medicines to kill bacteria and parasites can treat blood infections caused by these organisms. whoops, erythropoietin, a hormone produced by the kidney that stimulates red blood cell production. A manufactured form of erythropoietin can be given to improve the symptoms of anemia. Bloodletting. In people with problems caused by too much blood, such as from hemochromatosis or polycythemia, occasionally controlled removal of blood may be necessary. All right, I believe that that's all there is in this WebMD. So let's get rid of that. And this website is called redbloodcellbooks.com. Red Blood Cell Books, Breathe Knowledge. Red Blood Cell Factoids. So let's talk about the real red blood cells for a moment. In medical slang, they're called RBCs, but technically they're called erythrocytes. This unpronounceable looking word is the fusion of three Greek words, erythros meaning red, kaitos meaning hollow, and cyte, which we use for cell in the modern world. So RBCs are little red bags, and so they are, as you will soon see. RBCs are part of blood, which author Elaine N. Merrieb eloquently describes in her book Human Anatomy and Physiology as the river of life that surges within us, transporting nearly everything that must be carried from one place to another. Blood is made up of almost 45% RBCs and almost 55% plasma, a non-living liquid that the RBCs swim in. The not quite 1% left over are the germ-fighting white blood cells and platelets, the stuff that stops bleeding when something goes wrong with a blood vessel. Fun trivia. Blood accounts for 8% of your body weight. Fun trivia number 2. If we unraveled your entire circulatory system, it would stretch 60,000 miles. So back to our friends, the RBC. Our author William Lee Dubios describes red blood cells as the FedEx trucks of your body, moving oxygen from the lungs to the cells and carrying out the trash. He confesses that he's never seen the FedEx guy take out the trash, but you get the idea. RBCs offer both drop-off and pick-up services. They deliver oxygen from the lungs to each and every cell in your body, while there pick up the carbon dioxide waste and return it to the lungs for disposal. A red blood cell is really little more than a living bag of hemoglobin, a protein that oxygen grabs onto. Blood trivia. Red blood cells aren't really proper cells at all. The adult ones have no nuclei or organelles, crudely put, brain and guts, unlike most all other living cells. Red blood cells don't divide to reproduce themselves. They're built in the bone marrow inside your skeleton. Every day your body produces 100 billion new red blood cells. That's an ounce of blood, by the way. You produce 2 million per second, we're told. Baby red blood cells are blue. By cell standards, RBCs are small. A single RBC contains 250 million hemoglobin molecules. Each hemoglobin molecule can transport 4 molecules of oxygen. All that math adds up to each RBC can carry 1 billion oxygen molecules. RBCs look like miniature flying saucers, but they're more pancake than frisbee. They can twist, turn, and even roll themselves up like burritos to get through capillaries smaller than they are. RBCs don't use oxygen themselves. Crazy. Women have a lower red blood cell count than men do. When oxygen-rich, a red blood cell is described as scarlet or ruby red. When low on oxygen, the cell is dark red. The lifespan of a red blood cell is 120 days. Okay, so that was a quickie. On to the third. This one is from www.sciencedaily.com. And it's called How the Malaria Parasite Hijacks Human Red Blood Cells. It's dated July 10, 2008. The source is the Cell Press, and the summary says a new study done on a scale an order of magnitude greater than anything previously attempted in the field of malaria has uncovered an arsenal of proteins produced by the malaria parasite that allows it to hijack and remodel human red blood cells, leaving the oxygen-carrying cells stiff and sticky. Okay. Those effects on the blood cells play a major role in the development of malaria, a disease responsible for millions of deaths every year, the researchers report in the July 11th issue of the journal Cell. It's a nice piece of biology revealing how the parasite survives in and totally changes red blood cells, says Alan Coleman of the Walter and Eliza Hall Institute of Medical Research in Australia. Now that those players have been found, there may be some way of inhibiting these processes by drugs or possibly a live vaccine. Plasmodium falciparum causes the most severe form of malaria in humans, with 1 to 3 million deaths annually, the researcher said. Once in the blood, multiplication of the parasite inside the red blood cells, also known as erythrocytes, is responsible for its severity and mortality associated with the disease. After the parasite invades, the red cells undergo profound structural and morphological changes, dramatically altering their physical properties and impairing circulation. In contrast to normal red blood cells, parasitized cells are rigid and adhering to the lining of the blood vessels and other cell types. Those changes are known to be caused by proteins the parasite produces inside of the cell of its host and exports across several membranes out to the red cell itself. Earlier studies show two important ingredients: P. falciparum erythrocyte membrane protein, which allows infected cells to stick to blood vessels, and knobs made up of a second protein, knob-associated histidine-rich protein, or KHRP, that anchor PFEMP1 at the red cell surface. In the new study, the researchers were particularly interested in finding proteins required for trafficking PFEMP1 to the infected erythrocyte surface, correctly assembly of the anchoring knobs, and those involved in making infected red cells rigid, all processes associated with virulence and malaria infection, they said. In search of these players, the researchers developed mutant strains of P. falciparum, each lacking one of the 83 genes known or predicted to play a role in the red cell remodeling process. Their screen turned up 8 genes encoding proteins required for export of the PFEMP1 and assembly of knobs. Additionally, they show that multiple proteins play a role in generating increased rigidity of infected erythrocytes. In summary, the researchers wrote, "We have used a gene knockout approach on a scale not previously attempted in this organism to address the role of P. falciparum proteins that are exported into the parasite-infected erythrocyte. Collectively, these proteins act like the secretion systems seen in bacteria, in which pathogenicity arises from secreted proteins that interact with host cells by direct injection or by their presence in the extracellular milieu. It may be valuable to adopt approaches being tested in bacteria in which these systems are the target of new therapeutic approaches aimed at minimizing pathogen virulence." However, Kalman added, while they now know the identity of several proteins involved, they still don't know what these proteins actually do. Those are questions Coleman's team now plan to pursue in greater detail. The researchers include Alexander Mayer, the Walter and Eliza Hall Institute of Medical Research, Melbourne, Australia; Melanie Rugg, the Walter and Eliza Hall Institute of Medical Research, Melbourne, Australia; Matthew T. O'Neill, the Walter and Eliza Hall Institute of Medical Research, Melbourne, Australia; Monica Brown, the Walter and Eliza Hall Institute of Medical Research, Melbourne, Australia; Surbasti Chakravorty, Liverpool School of Tropical Medicine, Liverpool, UK; Taj Sestak, Liverpool School of Tropical Medicine. Okay, this whole rest of this paragraph is just all those names and stuff. I'm not going to go ahead and read all that. Story source: the above post is reprinted from materials provided by Cell Press. Note: materials may be edited for content and length. Cite this page, and right here, they cited it from the Cell Press, How the Malaria Parasite Hijacks Human Red Blood Cells. Science Daily, July 10th, 2008. So that's like almost 10 years ago, so I'm sure they have a lot different, a lot more information than that at this time. So, okay, done with that one. And the last one I have is phys.org. And this is dated April 16th of 2009. Researchers succeed in multiplying blood cells in the lab. Okay, a team from the Institute for Research in Immunology and Cancer at Université de Montréal has succeeded in producing a large quantity of laboratory stem cells from a number of blood stem cells obtained from bone marrow. The multidisciplinary team, directed by Dr. Guy Savagou, thus took a giant step towards the development of a revolutionary treatment based on those stem cells. This worldwide first will advance stem cell research and could have major implications in several fields for which no treatment currently exists. And I'm stuck. Every year in North America, nearly 4,000 people wait in vain for a bone marrow transplant due to lack of compatible donors. Whoa. What's going on with my page? It is known that a bone marrow stem cell transplant can reconstitute the recipient's bone marrow. The main difficulty is to obtain a sufficient number of compatible stem cells. Thanks to Dr. Savagou and his team, these patients will be able to obtain new bone marrow within the next few years. It could be possible to envision transplants for all adults from existing umbilical cord blood banks. The stem cell content of these blood banks is currently too limited for large-scale use in adults. Organ transplants without side effects, the medicine of the future. Currently, transplant recipients are condemned to take medications against rejection of the transplanted organ and suffer the side effects for the rest of their lives. However, mouse studies exist showing that bone marrow stem cells can prevent the rejection typically directed against solid organs. Rejection occurs because the immune system's cells manufactured by bone marrow attack the transplanted organ as if it were an invader. By extrapolation from laboratory studies, it is very likely that transplanting hematopoietic stem cells collected from the organ donor and developed in the laboratory could avoid rejection of this organ. This is why it's important to have large quantities of hematopoietic stem cells so the compatible stem cells can be matched with the organ to be transplanted. Using proteins to multiply stem cells. To produce large quantities of hematopoietic stem cells in the laboratory, Dr. Savagou's team identified 10 proteins out of 700 candidates. These 10 proteins are naturally present in hematopoietic stem cells, and researchers can use each of them to force these cells to multiply in the laboratory. The next step is to verify whether this also works in humans. Everything is already in place, Guy Savagou pointed out. These tests will be conducted at Montreal's Maisonneuve-Rosemont Hospital, one of the leading centers in Canada where stem cell transplants are performed. If only one of the 10 proteins allows hematopoietic stem cells to be multiplied in humans, we will be able to obtain the quantities of cells necessary to perform transplants. It will then be possible to say mission accomplished. Researchers around the world are currently trying to harness the regenerative power of other types of stem cells to treat diseases such as Alzheimer's or diabetes. IRIC's research could also help them achieve their goal. The work of Dr. Savagou's team has been funded by the Canadian Institutes of Health Research, and the findings are being published today at the prestigious scientific journal Cell. Source: University of Montreal. And that finishes this fourth website. Kind of interesting to dig up a topic and read 2 or 3 pages on it for no reason at all other than to learn something new.